Welcome to ChemSphere Hub. Today I will be presenting a comprehensive talk on factors affecting UV visible spectroscopy. UV visible spectroscopy is one of the simplest yet most powerful analytical tools used across chemistry, biology, nanotechnology, pharmaceutical sciences, and environmental analysis. However, the accuracy of UV visible results is highly dependent on several molecular, chemical, physical, and instrumental factors. Throughout this presentation, we will explore all major factors that influence spectra, how they cause spectral shifts, and how we can control these effects to obtain reliable analytical results. UV visible spectroscopy measures how molecules absorb UV visible light. As when light passes through a sample, certain wavelengths are absorbed due to electronic transition inside the molecules. This is the UV visible region. Ultraviolet region is from 200 to 400 nanometer. However, visible region is 400 to 800 nanometer. The resulting spectrum helps us to understand molecular structure, concentration, purity, and reaction behavior. However, because this technique is extremely sensitive, even small changes in the environment or sample conditions can significantly alter the results. Therefore, understanding the influencing factors is essential for correct interpretation. Now we will see the basic principle. UV visible absorption spectroscopy arises from the electronic transitions such as pi to pi star, n to pi star transition, and chi transfer transitions. Pi to pi star transitions occur in conjugated systems, and n to pi star transitions occur in molecules with lone pairs such as carbonyls. Each factor we will discuss today affects either the electronic energy gap or the availability of the electrons for excitation. This leads to change in lambda max, intensity, and spectral shape. UV visible spectroscopy plays a critical role in drug formulation and stability testing, nanoparticle characterization, determination of pollutant concentrations, reaction kinetics and mechanism studies, colorimetric sensing and biosensing, food analysis and industrial quality control. Because of such widespread use, proper understanding of the factors affecting UV visible spectra is essential for reliable scientific work. Now we begin with the molecular structure factors. These are internal factors controlled by the molecule itself. They include chromophores, functional groups responsible for absorption, conjugation. It is the extension of pi electrons, oxochromes, groups that modify absorption behavior, and molecular rigidity and planarity, and also the effects of electron donating and withdrawing groups. All these factors determine the intensity and wavelength of absorption. Now we will discuss the chromophores in detail, and chromophores are the structural unit that absorbs UV or visible light. Common chromophores include C double bond C, C double bond O, N double bond N, aromatic rings, and these chromophores define the baseline location of lambda max. More complex and extended chromophores typically absorb light of longer wavelength. Thus, chromophores are the primary reasons why molecules appear colored or absorb in specific region of spectrum. Effect of chromophores on the lambda max. Adding more chromophores or extending them lead to two major effects. In which one is bathochromic effect. In this, lambda max moves to a longer wavelength and creates the redshift. And the other one is hyperchromic effect. Absorbance intensity increases in this case. For example, benzene absorbs around 255 nanometer, but adding conjugation shifts absorption into the visible range, creating colored compounds like dyes. Now we will discuss the oxochromes. Oxochromes includes hydroxyl group, amino groups, sulfides groups that do not absorb strongly on their own, but they influence chromophores by donating or withdrawing electrons. They increase the intensity of absorption, hyperchromic effect, and shift lambda max either towards blue or red. This is why dyes consist of chromophores plus oxochromes. What is conjugation? Conjugation lowers the HOMO and LUMO gap. So it reduces the energy gap, and as more conjugation is present, it will reduce the more gap between the energy level, making it easier for electrons to be excited. As a result, lambda max increases, redshift will be present there, and absorption strengthens, spectra become more intense and defined. The degree of conjugation is one of the most important factors determining the color in organic molecules. Now we will discuss the molecular rigidity and planarity. Rigid and planar molecules show stronger electronic delocalization, giving sharper, stronger UV visible peaks. Flexible or twisted molecules have poor overlap of p orbitals, weakening absorption. Aromatic compounds like naphthalene, anthracene, and polycyclic aromatics absorb strongly due to their rigidity and planarity. Solvent effects. Solvent interact with the analyte, influencing electron distribution and energy levels. Solvent polarity, hydrogen bonding, and intrinsic solvent absorbance all significantly affect UV visible spectra. Solvent effects is very important, and it includes hydrogen bonding, solvent absorbance, and solvent interaction with other molecules, and also the solvent polarity. And we will discuss these factors in detail. Like, if we discuss the solvent polarity, polarity affects different electronic transitions. For N to pi transitions, polar solvents stabilize the excited state, causing a blue shift towards lambda max. And for pi to pi star transitions, polar solvents stabilize the ground state more, causing a red shift. It will lead to the higher lambda max. Therefore, solvent choice must be made carefully depending upon the type of compound. Now we will discuss the hydrogen bonding. Hydrogen bonding Now we will discuss the hydrogen bonding alters electron density and changes the environment around the chromophores. It typically broadens peaks, shifts lambda max. It may reduce absorbance intensity. Hydrogen bonding is most significant as hydroxyl amino groups and carboxylic groups are present there. Solvent cutoff wavelength. Each solvent absorbs strongly below a certain wavelength called its cutoff. For example, water absorbs at 200 nanometer, ethanol 205 nanometer, methanol 210 nanometer. Below these values, the solvent interference with the measurement is present. Therefore, solvent must be chosen such that the analyzed lambda max is above the cutoff value. Concentration effects. As the Baer-Lambert law states that the absorbance is proportional to the concentration. However, this is truly only at low to moderate concentrations. If the concentration is much higher, molecules will interact, they can aggregate or scatter light, leading to deviation. The optimal absorbance range for accuracy is 0.1 to 1. Deviations in Baer-Lambert law. Several reasons cause deviation in Baer-Lambert law, such as chemical deviations. They are due to association, dissociation, or reactions. Physical deviations are present like scattering, refractive index changes. Instrumental deviations like stray light, polychromatic radiation. Understanding these helps maintain linearity during quantitative analysis. Now we will discuss the path length. Path length affects absorbance directly because absorbance is equal to epsilon and concentration in path length. As most cuvettes are 1 centimeter, but special cuvettes of 0.1 centimeter or 2 centimeter are used for very concentrated or very dilute samples. So, longer path lengths give stronger absorption. So, length of cuvette will affect the absorption. So, it is a major factor. And cuvette material is also a factor which can be described. Cuvette material is extremely important. Quartz cuvette must be used in the UV region because glass absorbs strongly in UV region. Glass or plastic cuvettes can be used only for visible regions. Using the wrong cuvette leads to major inaccuracies, especially below 300 nanometer. As 300 nanometer, below 300 nanometer is the ultraviolet range, and in this case quartz cuvettes must be used. pH alters the protonation state of functional groups, changing the conjugation and the energy gap. Consequently, spectral changes occur. Indicators, for example, change color because their acidic and basic forms have different absorption maxima. Indicators like phenolphthalein can be discussed in this case. Now we will discuss the effects of acid-base equilibria. As acidic, basic and neutral species of same molecule often have different lambda max values, UV visible spectroscopy is frequently used to determine pKa values by monitoring these changings. This is especially important in pharmaceuticals and biological molecules. Temperature effects are very important, and they can be discussed here, as by increasing temperature increases molecular motion and decreases hydrogen bonding. This causes peak broadening, slightly lambda max shapes, decreased resolution. Therefore, temperature should be controlled in kinetic or thermodynamic studies. So, temperature is very important. Now we can discuss the instrumental factor. Instrument performance directly influence measurement accuracy. As if there is any problem with the instrument, then it will result in the measurement result. Factors: slit width, detector sensitivity, lamp stability, stray light and wavelength should be calibrated. They must be optimized for precise spectra. As these are the important factors which can be discussed, which should be optimized during a accurate measurement. And we will discuss these factors in detail. A slit width controls the amount of light reaching the detector. Narrow slit have higher resolution, sharper peaks. However, wide slit have more light, higher sensitivity but broader peaks. Choosing slit width depends on whether resolution or sensitivity is more important. Detector sensitivity. Detectors like photodiodes or photomultiplier tubes convert light into electrical signals. So lower sensitivity detectors produce noisy spectra, especially at low analyte concentration. So analyte concentration is also important, and detector sensitivity is important factor. Proper calibration and maintenance is essential in this case. Light sources also affects UV visible instruments. Use two main type of lamps: deuterium lamp that is used for UV region, and tungsten halogen lamp that can be used for visible region. Lamp aging, flickering or instability introduces noise and drifting baselines. So aging of lamp can also result in inaccurate results, and it can cause drifting signals. Wavelength calibration. Inaccurate wavelength calibration leads to wrong lambda max values. Calibration is performed using aluminum oxide filters or non-standard solution. This ensures correct and reproducible measurements. Any instrument can be calibrated by using the standard references filters. So wavelength can be calibrated. Scattering and particulates. This is also an important factor while describing the factors affecting the results in UV visible spectroscopy. In this case, particles, dust, bubbles or aggregated nanoparticles scatter light. Scattering increases absorbance artificially and distorts the baseline. So the sample should be particles free, dust free. Sample filtration or centrifugation is necessary, especially in case of nanomaterial studies. So they can cause false absorbance, and so filtration can filter the dust particles from the sample, and sample will then give the accurate results. And also the centrifugation is important so that the aggregated nanoparticles can be separated and the cleared sample can be analyzed, which can give the better results. Chemical interactions such as complex formation, dimerization, redox reactions and molecular aggregation significantly alter spectra. These interactions may produce new peaks or shift existing ones. So during the complex formation there can be chemical interactions present, and redox reaction, aggregation and dimerization, they can affect the results of the samples. Like metal ligand complexes, they often exhibit charge transfer transitions which produce intense colors and strong visible absorption. This is used to detect metal and study coordination chemistry. Lambda max also depends on the ligand field strength. This is used to detect metals and study coordination chemistry. Lambda max depends on ligand strength and metal oxidation state. Sample purity is also very important, as impurities introduce unexpected absorption bands and broaden existing peaks. Even small impurities can cause major changes in lambda max. High sample purity is essential for accurate UV visible analysis. So sample purity is important, as impurities can also introduce the unwanted peaks, as if the other molecules are also present in the sample, they will also give their peaks. They can do the broadening of absorption peaks, affecting quantitative analysis. In final, UV visible spectra are influenced by molecular structure, solvent environment concentration, pH temperature, path length, and instrumental settings. Controlling these parameters ensure accurate lambda max and absorbance values, leading to correct interpretation and reliable analytical results. A deep understanding of these factors is essential in research and industrial applications. So, in this lesson, we discussed the multiple factors influencing UV visible results. And we learn about the controlling of conditions for reliable spectra, to understanding the shifts that help in interpretation. So accuracy is very important if we use the samples like in research and industries. Thank you very much for your time and attention. Please like and subscribe my channel.